So, as we all know, biochars are the carbon-rich products of biomass pyrolysis used in carbon sequestration and as a soil amendment.
Do you
know where the volume is? Okay. All right, is that better? Okay. So when added to soil, biochars have been associated with a number of benefits. However, neutral and negative effects have also been observed. So given its rather broad definition, biochars can exhibit remarkable variety in their chemical and physical properties. This variability is largely due to differences in feedstock materials, pyrolysis conditions, mainly time and temperature, and post-production factors. The diversity of these materials is further enhanced by mixing feedstock materials together prior to pyrolysis. So the idea of mixing feedstocks stems from an interest in creating what has been termed designer biochars. These are chars customized to meet the specific needs of a soil, for example, rebalancing phosphorus concentrations or improving soil moisture retention. One salient characteristic of biochars is their sorptive capacity, which depends on their surface properties. Thus far, research has heavily focused on the sorption of pesticides and environmental contaminants in soil. However, naturally occurring compounds such as phenolic acids from root exudates and vegetative materials are also subject to immobilization. Phenolic acids are involved in a number of processes, including nutrient uptake, protein synthesis, and allelopathy. Their allelopathic effects are of particular interest given their potential use in weed management. So the phytotoxicity of phenolic acids depends on their bioavailability, persistence, and fate in soil. Because these compounds are only effective when they're in their free, unbound form, it's necessary to study their sorption. To date, a number of studies exist looking at the sorption of these compounds to soil. However, research on their sorption to biochar is still lacking. Of the published data, a number of studies looking at plant growth are looking at the interaction of phenolic acids and biochars. From these, we've learned that biochars greatly differ in their ability to disrupt the function of allelopathic chemicals, and certain biochars, when added to soil, can reduce the inhibitory effects of these compounds on both corn and asparagus seedling growth. Additionally, biochar can impact the mycorrhizal abundance and functioning, which can be negatively affected by phenolic acids. The objectives of our study were to characterize the influence of variation in biochar feedstock on the sorption of two phenolic acids, ferulic acid and syringic acid, as well as dichloric catechol. Secondly, we looked to determine how the sorption by biochars prepared from mixtures of feedstock materials differs from those prepared from the pure feedstocks. So the four feedstocks used in our study were switchgrass, swine solids, poultry litter, and pine chips. From each of these, a biochar was prepared, and these were considered our four pure feedstock chars. These same feedstocks were combined in various ratios prior to pyrolysis to give us four combined feedstock biochars. Switchgrass and swine solids were combined in an 80-20 weight-by-weight ratio, and pine chips and poultry litter were combined in 90-10, 80-20, and 50-50 ratios. All biochars, both the pure feedstock and combined, underwent pyrolysis at 350 degrees Celsius with a 2-hour residency time. We also included a Waukegan silt loam soil to compare sorption. This table outlines some of the main properties of the biochars. We had pHs ranging from 6.4 to 9.4, carbon content from 51 to 79%, and surface areas were all relatively low, ranging from less than 0.1 to 1.9 meters squared per gram. Here we see the structures of the three compounds. We have our two phenolic acids, ferulic acid and syringic acid, as well as dichloric catechol, which was included to determine the influence of a second hydroxyl group on the ring. All compounds were 14C labeled and purified using thin layer chromatography prior to use. So the sorption experiment was done using a standard batch equilibration method where half a gram of biochar or soil was added to a centrifuge tube along with 5 milliliters of a 1 part per million phenolic acid solution. The samples were then shaken, centrifuged, and the supernatant analyzed for 14C by liquid scintillation counting. To compare sorption, we calculated the sorption distribution coefficient, Kd, which equals the chemical sorbed divided by the concentration remaining in solution after equilibration. This value is then normalized to the biochar or soil organic carbon content to give us a KOC as well. This table shows the Kd values of our 4 pure feedstock biochars. For ferulic acid, Kd ranged from 1.4 to 75, syringic acid from 0.07 to 6, and dichloric catechol, as indicated by the asterisk, was completely sorbed on all but our poultry litter char. Looking at the 3 compounds, dichloric catechol was sorbed the strongest, followed by ferulic acid and syringic acid. So the sorption of these compounds to biochars is partly related to the chemistries of the compounds themselves. In soil, cinnamic acid derivatives have been found to sorb more strongly than benzoic acid derivatives, which also appeared true on the biochars in this study, with ferulic acid sorbing more strongly than syringic acid. Another characteristic of the compounds is their charge. So at the pHs of the biochars in this study, syringic acid and ferulic acid existed predominantly as anions, while dichloric catechol remained neutral based on their pKa's. So because biochar has a negative surface charge, the molecular phenolic group would be more readily sorbed than the anion. Additionally, in soil, compounds with a greater number of free phenolic groups have shown greater sorption, which again appeared true for the biochars, with dichloric catechol and its 2 free phenolic groups having the greatest sorption. And while both syringic acid and ferulic acid have a single phenolic group, the additional methoxy on the syringic ring could create steric hindrance, making this reactive group less accessible. So it's also worth noting that ferulic and syringic acid can rapidly react with iron and manganese oxides in both soils and biochars. However, we did not study the extent of this here. So the Kd and KOC values for the 4 pure feedstock biochars increased in the same order for both ferulic and syringic acid. The pine chip biochar had the highest Kd value, essentially the highest sorption, followed by poultry litter, swine solids, and switchgrass. Given this consistent order, we see that certain biochar characteristics are also influencing the sorption. However, we did not see any clear correlations between sorption and the measured biochar properties, which included pH, organic carbon content, and ash content. Furthermore, correcting for organic carbon content did not reduce the sorption variability, so our KOC values were just as variable as our KDs. So efforts have been made to identify trends between feedstock materials and biochar characteristics. To do this, they're often broken into groups of wood versus non-wood chars. The wood biochars typically have a lower ash content, lower pH, and higher surface area. The pine chip char in this study did in fact have a lower ash content and lower pH, however it did not have a higher surface area than the non-wood chars. Higher surface areas are typically associated with higher sorption. However, our pine chip biochar, which had the highest sorption, had the lowest surface area. For this particular char, the low surface area measurement could be due to resins, tars, or oils blocking the pore space or altering the surface properties within the pores themselves. So because biochar maintains the relic structure of the parent material, which can be so variable, so too can the pore distribution. Feedstock materials with large diameter cells, such as pine chips, lead to biochars with more macropores, which can readily absorb larger molecules such as phenolic acids. Unfortunately, surface area measurements don't often account for the size and shape of pores. Along with feedstock, surface area is also influenced by the pyrolysis temperature. Biochars prepared at higher temperatures often have an increased surface area. In our study, we did include one high-temperature biochar prepared from olive mill waste at 550 degrees Celsius, while all of the other chars previously mentioned were prepared at 350. This higher temperature char did in fact have a higher surface area, which could be responsible for its higher sorption of syringic and ferulic acid. So moving on to the combined feedstock biochars, in these graphs, on the y-axis we have our Kd value, which is essentially just the measure of sorption. And starting on the left, the x-axis shows the percent pine chip that was mixed with poultry litter to create the biochar. So 100% would represent our pure pine chip biochar, and 0% our pure poultry litter. A quick glance at the 50-50 blend shows that the KD value is not merely an average of its individual pure feedstock components. In fact, the 90-10 blend showed a lower sorption than either of the parent materials. Moving to the right, we have the percent switchgrass mixed with swine solids. And here we see the 80-20 blend had a higher sorptive capacity than either of its parent materials. So based on this, we see that we're unable to predict the sorption of combination biochars based on the amount of chemical sorbed to their pure feedstock components. This could be due to physical chemical alterations that occur during pyrolysis. For example, trace metal constituents can act as catalysts and alter the surface area during the pyrolysis process. So now looking at the soil, we saw a KD value for ferulic acid of 29, syringic acid had a KD of 12, and dichloric catechol 56. For these three compounds, we saw the same sorption order, with dichloric catechol sorbing the greatest and syringic acid the weakest. Now, comparing the soil KDs to the biochars, we saw that syringic acid was more strongly sorbed to the soil than any of the biochars studied. For ferulic acid, the same was true with the exception of our pine chip char. Comparing KOCs, the soil had a much higher KOC value than any of the chars. However, this was not particularly surprising given its lower organic carbon content compared to a biochar organic carbon. Again, mentioning that higher phenolic acid sorption to soil could be a result of mineral interactions. So because the biochars in this study sorbed ferulic and syringic acid less than the reference soil, the incorporation of these biochars into the silt loam would not likely alter the bioavailability of these phenolic acids to a large degree. These same biochars could, however, have a greater influence if added to soil with a lower sorptive capacity or in the presence of different phenolic acids. Again, phenolic acid structure, particularly the hydroxyl group, can impact its sorption to biochar along with the physical chemical properties of the biochar itself. However, we saw no clear correlations for this. Lastly, combining feedstock materials unpredictably affects the biochar sorption characteristics. Therefore, it's necessary to still study the sorption of these custom biochars. Thank you.